Hello, everyone, and welcome to today's webinar, ICH Q2 Validation of Analytical Procedures for Pharmaceutical Total Organic Carbon Analyzers. I am Michelle Ashton of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Beckman Coulter Life Sciences. To learn more, visit beckman.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing this presentation. This webinar is educational and thus offers free continuing education credits. Please click on the Continuing Education window at the bottom of your screen to obtain your credits. We have several poll questions that will be distributed throughout the webinar that will display on your screen. Your participation is greatly appreciated. I'd like to now welcome our speaker, Tony Harrison, Senior Marketing Manager at Beckman Coulter Life Sciences. Tony, you may now begin your presentation.
Thank you very much for that introduction, and welcome everybody to today's short talk on total organic carbon analysis for the Pharmaceutical Quality Control Laboratory and how it relates to ICH Q2. My name is Tony Harrison. I'm a Senior Manager at Beckman Coulter Life Sciences, and I will be your presenter today. Throughout this presentation, I'll be abbreviating the total organic carbon to TOC. So whilst this presentation is based around ICH Q2 and TOC, I've also included a little content around improving data integrity because I know it's such a hot topic for us in our industry, in the pharmaceutical industry today. Now, in their 2018 questions and answers document that the FDA published on 21 CFR Part 11, Data Integrity, they voiced their concern that they had increasingly observed cGMP violations involving data integrity during their inspections. And for the first time, they introduced the acronym ALCOA. I'm sure familiar to many of you, but it's used to describe what good data integrity looks like. And the last A in the acronym ALCOA stands for accuracy of the record's data. And what they're saying there is they're indicating that security and safety of the electronic record is important, but it's not the only focus of 21 CFR Part 11. It's also important the electronic record has accurate information or data in it. And here they're suggesting that anything that contributes to improving the accuracy of an electronic record is a positive thing. They always encourage the use of latest technology such as automation of the SOPs, which tends to reduce the impact of human error. Now, the QBD 1200 TOC analyzer from Beckman Coulter is designed to suit the pharmaceutical QC application. It can analyze TOC levels from 0.4 ppb through to 100,000 ppb or 100 ppm. It uses the industry standard combination of a UV light, ultraviolet light, and sulfate method. And its auto sampler can take up to 64 vials at a time. The user can program and automate their SOPs into the QBD 1200, which allows the operator to simply select the correct SOP for their testing using an intuitive icon-driven menu. So let's take a look at how the QBD 1200 analyzes TOC. In the first step of the analysis, the QBD acidifies the water sample being analysed to encourage any total inorganic carbon, or TIC, that's present in the sample to come out as solution. Now, the pharmacopoeias point out to us that TIC, or total inorganic carbon, tends to be in pharmaceutical grade waters purified water and water for injection is primarily made up from dissolved carbon dioxide gas. So after acidification, a carrier gas is used to purge the TIC out of solution and through to a non-dispersive infrared detector where it's measured and the total TIC is then reported. In the second step of the TOC analysis, a strong ultraviolet light is turned on to release the carbon atoms in the organic material present in the sample. The organic material is oxidized by hydroxyl radicals which are created by the effect or the impact of the UV light on the water molecules themselves. Plus then the UV light also combined with the sulfate activates the sulfate, and the TOC is released from the organic as CO2 gas. It's important to note that sulfate doesn't actually act as an oxidizer until exposed to the UV light. In the third analysis step, the carrier gas is used to sweep the CO2 from the oxidized TOC out of solution, and it is then measured by the non-dispersive infrared detector. Now, as I mentioned a few moments ago, in their 2018 questions and answers documents on 21 CFR Part 11 data integrity, the FDA voiced their concern that they were increasingly observing GMP violations involving data integrity during their inspection, and they indicated that whilst the security of the final electronic record is important and it should be protected against adulteration, it's not the only focus of 21 CFR Part 11, but also that the electronic record contains accurate data too. Now, they introduced the acronym ALCOA in their 2018 guidance, and it stands for attributable, legible, contemporaneous, and original. And the last A in the acronym stands for accuracy of the electronic records data. And as I say here, they're saying that anything that contributes towards improving the accuracy of the electronic record, the data it contains, is a positive, such as automating SOPs. Now, a 2016 report states that 79% of all of the warning letters written by the FDA, their 483 letters, in that year cited 21 CFR Part 7 data integrity issues, so 79% of the letters. And whilst retraining is a common response to an FDA 483 warning letter, this may be only part of the solution. Was the problem simply that the workers didn't follow the SOP correctly, or was it really that manual SOPs are simply not robust enough to prevent future recurrence of the problem? Now, any process that automates the quality control SOP can potentially both reduce the impact of human error on data integrity issues and also brings welcome savings by reducing the time technicians need to use to devote to carrying out the SOP itself. Now, here's an example where manual SOPs can lead to error and inaccurate records. In some commonly used QC lab TOC analyzers using carbon dioxide permeable membranes as part of the measurement process, the users, the technicians, have to guess how much oxidizing persulfate to add. In other words, they need to know the level of TOC present in the sample before they carry out the test. But how can they know this? So the problem is that if they add too much persulfate, more than is required for the level of TOC present, you can get bubbles forming in the analysis chamber, and they block these permeable membranes that are used in some analyzers, and this causes the analyzer to underreport because the carbon dioxide can't get through the membranes and be measured. Equally, however, if they don't add enough persulfate for the level of TOC present in the water sample, the TOC will not be fully oxidized, and again, the TOC analyzer will underreport. Now, the QBD 1200 from Beckman is not affected by bubbles in the sample. And as a minimum, and by standard default, the QBD 1200 adds sufficient sulfate oxidizer to each sample to safely oxidize up to 4,000 ppb of TOC. So this ensures that TOC results are not underreported in normal water samples like purified water and water injection. All TOC analysis SOPs can be pre-configured and programmed into the QBD 1200, so the technician has reduced manual configuration steps, which helps to reduce the opportunity for human error, which of course could affect the accuracy of the data in the electronic record itself. So let's now move on to ICH Q2 in the second part of our presentation today. The primary purpose of the Tripartite International Conference on Harmonisation, ICH, their committee is the primary purpose is to harmonise the Japanese, United States and the European pharmacopoeias. However, they also perform other functions such as producing guidance such as the ICH Q2 document, which is about validating instruments to make sure they're fit for purpose. There's an IQ, OQ and PQ documented procedure available for the QBD 1200, and the IQ, OQ sections, of course, confirm that the analyzer is installed and performing as intended, but the PQ section carries out the required testing to validate the analyzer to the QBD to ICH Q2 guidelines. The ICH Q2 document provides a discussion of the characteristics for consideration during the validation of an analytical procedure, including those listed on the slide. So let's take a look at each of these attributes in detail and how the PQ from the QBD 1200 demonstrates compliance. ICH Q2 suggests that an instrument's accuracy can be demonstrated by examining the instrument's response to a certified reference standard, so a certified traceable reference standard. The QBD 1200 PQ uses a certified potassium hydrogen phthalate solution containing 5,000 ppb or 5 ppm of TOC. Automated onboard dilution allows comparison against this standard at 5 points across the measurement range. The test SOP is preloaded and automated and is selected from an icon on the screen. So there's no manual configuration, no manual pass-fail, so it reduces the opportunity for human error. Now, although the United States and the European pharmacopoeias do not specify the calibration solution to be used on TOC analyzers, but the Japanese pharmacopoeia does, it calls for KHP to be used. And hence, by using KHP, the QBD 1200 can be seen to comply with all three pharmacopoeias. The QBD 1200 accuracy is demonstrated by comparing its response to 5 different levels of TOC. And the pass-fail calculation is automated, so there's no manual calculations of the pass-fail. And this avoids them and reduces human error. Now, ICH Q2 goes on to suggest that precision of an analyzer can be assessed by comparing multiple analyses of the same sample. It recommends 3 replicates of 3 concentrations of TOC are used, and the PQ for the QBD 1200 follows this guidance by doing 3 replicates each of 1, 2, 3, 4, and 5 ppm or 5,000 ppb of TOC. The performance of the QBD 1200 against this precision test is calculated automatically, providing the results of a standard deviation calculation. Now, in this slide, we're explaining the difference between accuracy and precision. I'm sure most of you are familiar with this, but the first dartboard at the top left there shows what happens when you have neither accuracy nor precision. So all the hits on the dartboard are all over the place. The second dartboard on the top right shows excellent precision with very repeatable results, but it's nowhere near the bullseye, so it's got poor accuracy. Now, the third dartboard shows relatively good accuracy with the results clustered around the bullseye, but the precision or repeatability of the testing is poor, as the results are not consistent. And finally, the fourth dartboard shows us what both good accuracy and high precision combined together looks like, with the results repeatable and on target. Now, the specificity test listed in ICH Q2 demonstrates that the TOC analyzer can measure and report TOC accurately, both over the range of expected TOC substances to be found in typical pharmaceutical waters, and also in the presence of potentially interfering substances that may be in the sample. Compliance with the first part is demonstrated by carrying out the system suitability test to ensure that the TOC analyzer can successfully analyze the full range of organic materials expected to be present in pharmaceutical grade waters. The ability to successfully measure measure TOC in the presence of large amounts of potentially interfering inorganic carbon, or TIC, is demonstrated by challenging the analyzer with two samples. They both contain the same amount of organic carbon, but one of them contains large amounts of inorganic carbon in the form of dissolved CO2, which is typically how you find purified water. So if the analyzer can report the same TOC results from both samples, then it's deemed to have passed successfully. The QBD 1200 can handle very high levels of TIC because it completely sparges it or pushes it out from the sample before attempting to measure TOC. Once the TIC is sparged from the sample, the UV lamp is turned on and the analysis of TOC commences. The QBD therefore makes a direct measurement of TOC, not a calculated one. So it actually measures TOC directly, not calculating it. And so the plus or minus 2% tolerance on the sensor that the QBD uses means, for example, that there might be potentially a plus or minus 2 parts per billion potential error in a sample containing 100 ppb of TOC. And this is despite the presence of relatively large amounts of TIC. Now, the limit of detection is the smallest amount of a substance the analyzer can detect, but not necessarily accurately report. Whereas the limit of quantitation is the smallest amount of a substance that the analyzer will not only detect, but will accurately measure and report. And neither of these, of course, is very easy to do in the real world, especially with a limit of detection of 0.4 ppb. So what ICH Q2 does is provides guidance on how to calculate the limit of detection and limit of quantitation based on the standard deviation of the results from a calibration. Now, the linearity test defined in ICH Q2 determines the ability of an analyzer to report accurately over a range for analysis. And the performance qualification for the QBD shows this by across a range of measurements. So the linearity test pass-fail calculations, automated, and again avoids manual calculations. And the range of the QBD 1200 is demonstrated by testing in the measurement range and then explaining that the higher levels of TOC are analyzed through automatic sample dilution using a calibrated syringe pump accurate to plus or minus 1%. So in the case of TOC, 1 part per billion is equivalent to 1 microgram per litre of water. And of course, a litre of water is a kilogram. If it's pure water, weighs a kilogram. So it's important to understand, because 1 litre sample containing, say, 100,000 parts per billion of TOC, which is 100 micrograms per litre, if that's then diluted by another litre of TOC-free water, so water with no TOC in it, The TOC level in the resultant 2 litres of water is halved and is now just 50,000 ppb. So if you dilute the water sample under test, you reduce the ppb levels of TOC present. By employing this dilution technique, the QBD can dilute and analyze water samples containing up to 100,000 ppb of TOC, even though the actual measurement range of the non-dispersive infrared detector is only 5,000 ppb, i.e. the QBD dilutes the sample by a factor of 20, and of course 20 times 5,000 gives you the 100,000. In samples up to 4,000 ppb, TOC sample dilution is not required, as the amount of sulfate added automatically each time by the QBD will successfully analyze this level of TOC. Above this level, the QBD automatically determines the amount of sulfate to add, and so the user does not need to guess. So this is one of our first poll questions. So we'll now move on to robustness. Now, robustness, according to ISPE Q2, is the ability to measure successfully despite variations in the sample characteristics. Now, as the QBD adds acid to the sample to encourage the CO2 to come out of solution and be measured as a gas, in theory, the pH of the sample might impact on the measurement. However, as part of the performance qualification, it demonstrates this is not the case by deliberately testing the QBD with samples of TOC where the pH has deliberately been altered, showing that the measurement performance of the QBD is not affected by shifts in the pH. So this is our next poll question. Okay, moving on to intermediate precision. Now this is an interesting one, and it's really looks at the effects of random events on the precision of the analytical procedure itself. So it's looking at typical things to study, such as time of day, temperature, but also the technician or analyst who's using the TOC analyzer as well. So the way we demonstrate this with the PQ is that we automate the SAPs, so the instrument's automatically configured the same way every time. And also we automate the addition of sulfate to ensure the TOC is analyzed correctly. So we don't need technicians who can guess how much TOC is in the sample and therefore how much sulfate to add. It's all automated. And finally, the performance qualification demonstrates its robustness by being unaffected by gas bubbles in the sample because the NDR detector is selective to only measure carbon dioxide. The standard amount of sulfate added covers even the widest range of TOC analysis required in a pharmaceutical grade water system. So that concludes our short presentation today. I hope you found it informative. I'll now take any questions the audience has, please.
Well, thank you, Tony, for your informative presentation. We will now go ahead and start the Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many questions as we have time for. So let's go ahead and get started. Our first question, you mentioned 21 CFR Part 11 data integrity. Could you please explain again how many error can lead to data integrity challenges?
For sure, definitely. So yeah, human error, if it creeps in the test, in the configuration of the instrumentation, it can cause problems with data integrity. Now, in the FDA's ALCOA acronym, as I mentioned in the presentation, the final A in that acronym refers to the accuracy of the electronic record itself. So we're not talking about security or anything like that. It's really about the data in the electronic record. So human error in TOC analyzer configuration can lead to errors in the analyzer results. So the electronic record itself, although secure, is not accurate. It doesn't represent an accurate analysis. So I gave an example of how human error can creep into TOC measurements just now when I explained how users of some typical TOC analyzers have to guess the amount of TOC in the water sample in order to dose the correct amount of sulfate. So the temptation is not to put too much sulfate in, because that runs the risk of creating bubbles and under-reporting the TOC in the sample. But that potentially leads to accuracy issues in the electronic record. Unfortunately, the QBD measurement is not affected by this problem.
Thank you, Tony. Our next question. You mentioned that with some TOC analyzers, the technician has to guess how much of oxidizer, the persulfate, to add to the sample based on a guess of the TOC content in the sample. But how can they guess the TOC content before they make a measurement?
That's exactly the point, really. It's kind of a chicken and egg thing, if you excuse the analogy, because you have to know how much TOC is in the sample to add the persulfate to then measure the TOC. So it's a challenge, and it faces many technicians using TOC analyzers today. But unlike some commonly used QC lab TOC analyzers that measure organic carbon dissolved in the water sample using permeable membranes as part of the measurement process, which is what causes the issues with sulfate, the QBD 1200 measures carbon dioxide from the organic carbon in gas phase and doesn't use permeable membranes. So the presence of gas bubbles does not interfere with the measurement. Hence, when we're measuring, whether we're measuring purified water or water rejection, we can safely add enough the sulfate every time to analyze up to 4,000 ppb of TOC without it interfering with the measurement in any way at all. And of course, as TOC is a limit test, with the maximum limit being 500 parts per billion, the ability to oxidize to 4,000 ppb ensures that any excursions above the 500 ppb limit would be captured and reported.
Thank you, Tony. We have one more question here for you. Thank you for your presentation. It is well known that some laboratory TOC analyzers measure both inorganic carbon and total carbon using separate sensors and then use these two measurements to calculate TOC, frequently resulting in errors in the reported TOC value, even sometimes reporting negative TOC results due to sensor drift. Could you please explain how the QBD 1200 avoids these types of errors?
Thank you. This is a good question. And I think the basic is that unlike some laboratory TOC analyzers, the QBD 1200 removes the inorganic carbon from the sample first and measures that directly. And then it oxidizes the TOC and measures the carbon dioxide from that. So it's directly making a measurement of TOC using a single sensor, which has an accuracy of plus or minus 2%. So because it's using a single sensor instead of two different sensors, it's not possible for the QBD 1200 to report a negative TOC result because it measures TOC directly rather than calculating it from two separate measurements.
Well, thank you, Tony. Do you have any final comments for our audience before we close today?
No, just thank you very much for attending and listening to my presentation. I hope you thought it was useful, and enjoy the rest of your day.
Thank you again, Tony, for your time today. We would also like to thank LabRoots and our sponsor, Beckman Coulter Life Sciences, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Reminder, questions we did not have time for today and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues or anyone who may have missed today's event. Until next time, goodbye.
Goodbye.